Hello, my name is Ayesha Ullah, and I will be presenting my research on the confirmation of the presence of Acanthamoeba species with Mycobacterium leprae from soil samples that were collected at the Yazoo Wildlife Refuge. And so a little background on what Acanthamoeba and Mycobacterium leprae, or M. leprae, are. Acanthamoeba is actually a type of amoeba that is free-living and can be found in freshwater habitats as well as in soil. And some species are known to be parasitic to humans and cause infections in areas such as the eyes and the skin. And so Mycobacterium leprae, or M. leprae, is a type of bacterium that carries leprosy, and this can be found in areas where leprosy is endemic to humans. And so I chose to focus on Acanthamoeba and Mycobacterium leprae because previous research has shown that M. leprae is able to survive up to 35 days in Acanthamoeba, in Acanthamoeba as well as Hermantella, which are both free-living amoebae species in beets. And so humans and armadillos in southern regions of the United States are being infected with identical M. leprae strains. And as right now, researchers are trying to find out whether or not Acanthamoeba species serve as possible reservoirs for M. leprae in the soil where leprosy is endemic. And so in order to determine this, we took isolated genomic DNA from the soil samples that were collected and assessed them for the presence or absence of M. leprae through PCR amplification, and then the sequencing of those PCR amplifications. And so for this study, 41 genomic DNA isolates were studied, where 23 were M. leprae positive samples, and then 18 were M. leprae negative samples. And then they were tested for Acanthamoeba using PCR amplification and then gel electrophoresis with positive and negative controls. And then for our areas of focus, I chose two research questions, one being, is Acanthamoeba present in soil samples that were positive for M. leprae? And then two, is Acanthamoeba present in soil samples that are negative for M. leprae? And then determining whether or not there's a relationship in the presence of Acanthamoeba and in the presence of M. leprae. And so based off of the results I gathered from the gel electrophoresis, only one out of the 41 samples that were done did not have Acanthamoeba in it. Did not have a presence of Acanthamoeba, and this was sample B22, and this was determined based off of, so in each gel electrophoresis, a positive and a negative control was used, and for example, we see that B22 does not have anything in it where the negative control is, so therefore it did not have a presence of Acanthamoeba. And so based off of the fact that 98% of the samples did have a presence of Acanthamoeba, regardless of whether or not they were positive or negative for M. leprae, I determined that there is not a correlation or a relationship between the presence of Acanthamoeba and the presence of M. leprae, since Acanthamoeba was found in samples that were positive and negative for M. leprae. And so based off of this, I determined that there is a limitation in the study in that the samples that were collected are very minute compared to the samples that were gathered from the refuge, and therefore there could be traces of M. leprae or Acanthamoeba that were not detected through our testing, and so further testing would have to be done, as well as additional samples. I only did 41 samples. Additional samples could be done in order to further determine the relationship between Acanthamoeba and M. leprae. And so in conclusion, I would like to thank Dr. Gardner and Dr. Andrus for their support through this research project, and as well as the Meredith Department of Biological Sciences that supported me in this project that was done in collaboration with Valdosta State University. Thank you.